Welcome to LearnQuest Academy YouTube channel. Today we are going to be doing the predictions made by LearnQuest Academy for the Biology WASSCE exam part 2. I want to start with a disclaimer. Please note that this is not Expo. It is simply the product of the careful analysis made by the tutors at LearnQuest Academy to help students pass their Biology WASSCE exams excellently. Lest I forget, make sure you follow the LearnQuest Academy on WhatsApp. You gain a lot from it. Wishing you success in your exams. The first question I have here is, state 5 differences between mitosis and meiosis. Why did I pick this question? WAEC sets this question, or rather, WAEC sets questions on mitosis and meiosis every year. It can appear in either or both of objective and theory. WAEC also set a similar question to this in 2023, so it's also a recent question. The analysis performed by WASSCE in 2008, question 4, was a question on mitosis and meiosis, and it revealed that candidates who attempted it generally performed poorly. Just like I said in my previous video, WAEC will constantly set questions that candidates will perform poorly because they are an exam body, and not every exam body wants a student to pass excellently. Many of the candidates who attempted this question 4 in 2008 just stated the stages. They do not describe the processes. The question was states the stages of mitotic cell division, or rather the question was explain the process. Yes, explain the process of mitotic cell division, but they only stated the stages. They did not describe the processes. So it's very important that you know the processes. I personally did my own analysis and I found that many students actually struggle with mitosis and meiosis. To answer this question, we have to understand exactly what each of mitosis and meiosis entail. Mitosis is a cell division that involves the division of the parent cell into 2 diploid daughter cells, and each has the same number of chromosomes as the parent cell. Take note of the keywords: diploid, same number of chromosomes. We have 4 major stages of mitotic cell division, which is the prophase, the metaphase, the anaphase, and the telophase. In the prophase, the chromosomes shorten, thicken, and become visible. The spindle fibers are formed, and the centrioles move to the opposite pole. In metaphase, the nuclear membrane disappears. Just like you can see here, there was a nuclear membrane in this place, then disappeared. So the nuclear membrane disappears. The chromosome line up at the equator. If you did geography, you are familiar with equator poles. So this is the equator. Even if you didn't do geography, if you did maths, the chromosome line up at the equator and attach themselves to the spindle fibers using the chromosome. Now must have been a mistake. They attach themselves to the spindle fibers. These are the spindle fibers, and the chromosome attach themselves to them. In the anaphase, the spindle fibers contract and pull the chromosome. The sister chromatids separate. Each chromatid moves towards the opposite pole along the spindle fiber. It finally separates. And the last stage, telophase, in which spindle fiber, you can't see them again. They've disappeared. Then nuclear membrane forms around each set of chromatids at each pole to give 2 new daughter cells. After the chromatids went to the poles, nuclear membrane quickly formed around them, and cytokinesis takes place, which is the division of the cytoplasm. There is one more crucial stage in mitosis, that is the interphase. Although it is not a process in mitotic cell division, it's a crucial phase after each mitotic cell division. It is a stage of synthesis of body materials in the daughter cell after each successful cell division. The chromosome appear as thread-like materials in this stage. I'm sure that if you've seen the drawing of chromosome in other areas of biology apart from cell division or genetics, you see it like this. Yeah, so that's the thread-like material that we're referring to. In interphase, this is how the chromosome look like, not this way. Replication of chromosome to form two chromatids. Look at this. Before we had this, now we have just this. It is in interphase that this one will form this. I hope you get. Lastly, cell enlargement and preparation for the next cell division takes place in interphase. Here is a simple way to remember the processes of mitosis in order. Prophase, pro stands for before. When you say proactive, you are preparing before the danger eventually takes place. Meta stands for next, so it's the next phase after prophase. Anaphase stands for back or up, where the chromosome pull apart from each other or pull up from each other. The telophase, telo stands for end, and it's the last stage. In case you don't remember what ana, telo, meta, or pro means, you can use this PMAT. If you are fine with just cramming PMAT, it's fine. Or, these are the processes of mitotic cell division. So P stands, processes as a P, and that's the P. Mitosis as a M and a T. So definitely A will be between M and T, if you get what I mean. So this is also a simple way that you can use to learn it. Processes of mitosis. Moving on to the processes of meiosis. Meiosis is a cell division that a diploid parent cell divides to form 4 haploid daughter cells, with each having half the number of chromosomes at parent cell. If you remember what we said about mitosis, it is production of 2 diploid. Now meiosis is leading to 4 haploid, and if it is 4, that means that definitely something must go for it, and that thing that went for it is the number of chromosome. The number of chromosome reduced to half. A way that you can always use to remember that haploid means half the number of chromosome. You know that haploid, it sounds something like half. If you get, it sounds like half. So haploid sounds like half, and haploid means half. So 4 haploid daughter cells. There are 2 major stages in meiosis, which is the first meiotic cell division and the second meiotic cell division. You also understand why we have to have 2 meiotic cell division. In the first meiotic cell division, the homologous chromosome separates, while in the second, the sister chromatids separate, just like mitosis. Homologous chromosomes do not separate in mitosis. There are no homologous chromosomes in mitosis, or rather, there are homologous chromosomes, but they do not separate. In the first meiotic prophase, the homologous chromosome pair up. I used large and small homologous chromosome to represent the homologous chromosome. So these small ones are homologous. Homo means the same or similar. So these small ones are homologous, and this large one are also homologous. After the homologous chromosome pair up, as they touch each other and come in contact with each other, there is crossing over of genetic materials. After the crossover and exchange genetic materials, the centrioles split and move over each pole. These are the centrioles. These two dots or two circles. This is the spindle fiber which is formed. Before I move on, later in this video, you understand the consequence of the crossing over of genetic materials. The first meiotic metaphase involves the chromosome lining up at the equator and the nuclear membrane will disappear. In the anaphase, the homologous chromosome separates and each chromosome moves to the respective opposite pole of the cell. After that, the telophase, cytokinesis, definitely. There has to be division of the cytoplasm. Each chromosome goes to each pole of the cell. The spindle fibers disappear. Cytokinesis occurs to form two daughter cells. Then nuclear membrane forms around each set of chromosomes. In the second meiotic cell division, the centrioles split, almost like mitosis. The centrioles split and move to the poles. Formation of spindle fibers also occurs. In the metaphase, same thing as mitosis, line up at equator, nuclear membrane disappears. Anaphase, same thing as that of mitosis, spindle fibers contract, chromatid separates, each chromatid moves to the respective pole. And the telophase, same thing as mitotic telophase. The nuclear membrane forms around each set of chromatids at the respective poles. The spindle fiber disappears, it has to go. The cytokinesis occurs. Definitely there has to be division of the cytoplasm. And the four haploid daughter cells are finally formed. Now that we understand what mitosis and meiosis entail, let's look at the question. The question is differences between mitosis and meiosis. Mitosis occurs during growth and asexual reproduction, and that's why it occurs in the somatic cells. Somatic cells are body cells. So your hand, your brain, and your legs. This is mitotic cell division that occurs there. Meiosis occurs in the germinal or germline cells. Let me type out germinal so you can understand what I mean. Germinal. Meiosis occurs in the germinal or germline cells or gonads, and it occurs during gametogenesis or during sexual reproduction. In mitosis, two daughter cells are formed. In meiosis, four daughter cells are formed. There is no crossing over of genetic materials in mitosis, and there is crossing over of genetic materials in meiosis. I said I was going to explain the consequence of this. Since there is no crossing over of genetic materials in mitosis, the daughter cells will be identical. But since there is crossing over in meiosis, daughter cells will show variation, because that means that the chromosomes do not have the exact same composition of genetic materials, which will lead to variation. But in mitosis, they are identical. In mitosis, each daughter cell has the same number of chromosomes, as evident in the definition. But in meiosis, they have half the number of chromosomes as the parent cell. In mitosis, there is no pairing of homologous chromosomes. But in meiosis, there is pairing of homologous chromosomes. In mitosis, the nucleus of the cell divides how many times? Once, because there's only one mitotic cell division. But in meiosis, there are two stages of cell division, so the nucleus divides twice. Ecological succession is the next question. Explain ecological succession. I picked this question because This question was last tested in 2013 and it's been a while, so WAEC may bring it up again. And they also tested it a lot of times, 1989, 2002, 2006, 2008. So they may bring it up again. Ecological succession is the gradual replacement of old species by new species. Remember that succession comes from the word succeeding. When you succeed someone, that means that you take over what the person used to take over before. Do you get? So ecological succession comes from the word succession. You might be wondering what a climax community is. A climax community is also a very important definition you should take note of. It's a community that is in equilibrium with the environment so that there is no more succession and it remains the same in composition and size over a long period of time. It can also be defined as the last stable stage of ecological succession. But if you are asked in your exam, I do not advise that you define it in this way, last stable stage. Just define it as a community that is in equilibrium so that there is no more succession and it remains the same in composition and size over a long period of time. Just like I said in my previous video, you don't have to cram definitions. Just understand what the definition means, then note the keywords. In this case, it is equilibrium, no more succession, same composition, and you're good to go. It will be very easy to remember the definition that way. Types of succession: primary and secondary. Let's differentiate between primary succession and secondary succession. Primary succession occurs when there was never a community before. Secondary occurs when there was a community originally and it occurs in a disturbed terrestrial habitat. So maybe there was a forest fire, that's a disturbed terrestrial habitat, and there was a community, definitely a community there before. For forest fire to occur, there has to be trees, there has to be animals there. Primary succession takes a longer time, hundreds to thousands of years, while secondary takes a shorter time to occur. In primary, there is no soil present before it starts, so it involves soil formation. But in secondary, soil is already present before it starts. In primary, pioneer organisms. 4 is the same thing as 1, so I do not want to repeat number 4. In primary, we have various kinds of pioneer organisms, but the most common ones are lichens on rocks. Lichen is the relationship between algae and fungi. So lichen is majorly algae. Lichen on rocks, red mangrove on muddy river mouths, phytoplanktons on ponds. Phytoplankton comes from phyto, and phyto comes from chlorophyll. So phytoplankton are plankton refer to microscopic organisms or very, very tiny organisms. So phytoplanktons are very tiny plants. For example, Chlamydomonas is a phytoplankton. In secondary succession, pioneer organisms are plants from the previous climax community. So in case of a forest fire, there were plants there before. The pioneer organism can still be some plants that were formerly in that community before it was disturbed, or from surrounding community. And pioneer organisms in primary succession refer to the first organisms, while pioneer organisms in secondary succession refers to the organism that succeed the organisms that were present in the habitat before the habitat became disturbed. In my previous video, I said I was going to explain what the various views in a fish, or rather the various views that we have in biology. I'm using a fish as an example, and I got this from Wikipedia Commons. This is the lateral view of the fish, which is also the side view. So in case of a human, the lateral view would be the hand, where your hand is. So that's the lateral view. The central or anterior view, if you are asked in the exam, they might not say anterior view, they might say central view. So you have to know what this means. Or the front view, or the anterior view. In humans, that would be looking at the top of your head. But in a fish, it is looking straight into the eyes of the fish. different kind of organisms have their different views. So a human being now does not look like this. The face is like this. This is where the face is. Then the hands are here, their legs. So you cannot compare the anterior view of a fish or the dorsal view of a fish to that of a human. The dorsal view of a fish is the top view. In human, that would be the back of the human. So we will be looking in human, we'll be looking at your spinal cord. That will be the dorsal view of a human. The posterior view of a fish, you are looking from the back of the fish. This is the fin of the fish here. But in human, it will be like you are looking from the bottom of your leg. The ventral view of a fish is this, which is the bottom view. In human, it will be your belly. That's your ventral view. Your belly, your face, your belly downwards. The cross section of a fish is you are cutting the fish this way. And this is how the cross section or the transverse section looks like. While the longitudinal section is also the lengthwise section. And you can get a view from cutting the fish this way downwards. Thank you for watching to the end. This is a reward for watching to the end. I compiled the acronym for the major elements of plants that are derived from the soil. Plants operate or survive on major elements. So these are the elements that are derived from the soil: nitrogen, phosphorus, potassium, sulfur, calcium, magnesium, iron. Please take note that this reward will not be put in the note I'll be releasing. Yes, and that reminds me, the notes for the notes I am using to teach you right now will be released in the video description. Do also check it out. But this reward for watching to the end is only available on this video, so you might want to write it down. The acronym for the major element of plants is: nourished plants keep showing consistent massive increases. This is an easy acronym to know because definitely nourished plants are plants that are healthy, so they will definitely keep showing consistent massive increases in their crops or in their yield. The elements are nitrogen, phosphorus, potassium, NPK, then sulfur, calcium, magnesium, and iron. Nourished stands for nitrogen. What does nitrogen do? Nitrogen functions in protein synthesis. If you've done chemistry, you will know that most proteins contain nitrogen. So nitrogen is an important element in plants. Phosphorus is for energy transfer. You know ATP. ATP is for energy. In plant and animals, all organisms use ATP. ATP is adenosine triphosphate. So phosphorus is for energy transfer. Potassium is for water or turgor regulation. It's a very strong electrolyte and a very reactive metal. So it's very good in plants for water regulation. Sulfur is for protein synthesis because most proteins contain sulfur. Calcium is for cell wall formation. C for calcium, C for cell wall. That's a very easy way to remember. Magnesium is for chlorophyll formation. Magnesium and iron are for chlorophyll formation. Take note of that. I could not derive an acronym to help you always remember it, but this is a very common question in WASSCE. Which of the following or which set of the following are for chlorophyll formation? Which of the following set of nutrients are for chlorophyll formation? And it is magnesium and iron. I asterisked this iron because although for this stage iron is classified as a major element in most textbooks, but it's actually a minor element because it's required in very small quantities. But if you don't have iron, you cannot produce chlorophyll at all. So that's why some textbooks for this stage classify iron as a major element. Thank you so much for watching to the end. Don't forget to like and subscribe for more quality content like this. See you later.